This is a muscle contraction video. Is it recording audio? It has to be recording audio. Action potential.
Now I have to start over.
Okay, how do I do that?
Okay.
Alright. We can edit it out. We can edit it out.
Do you
It's
know how
going.
to edit it?
Oh, we're gonna have to learn in a few minutes here.
Okay.
Alright. So the action potential arrives at the synaptic knob, calcium channels, and then the synaptic knob's right there, the purple.
Right there.
Stop. Go. The calcium channels in membrane open up, allowing the calcium to move into the cell. We have everything color coordinated here, so. The increased calcium causes the synaptic vessels to fill with acetylcholine to fuse with the membrane. The acetylcholine is released from the synaptic knob by exocytosis into the synaptic cleft. The acetylcholine is picked up by receptors on the motor end of the skeletal muscle, or the motor end plate of the skeletal muscle. Sodium-potassium channels open to allow the sodium ions to enter muscle fiber. Increase in ions inside muscle cell initiates an action potential on the sarcolemma. APT travels down sarcolemma into... Sorry. My talking and her hand coordination are not in sync at this moment.
What did you say? ATP what?
Now, the ATP travels down the sarcolemma into T tubules and stimulates the release of calcium.
You're over here.
So that was the stimulation of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it has been sequestered by the calcestrine into the cytoplasm of the muscle cell. Calcium floods over the thin filaments and binds to the troponin, cooling tropomyosin. Off the active site on the actin. High energy myosin heads leap to bind on the exposed active sites on the thin filaments, forming a cross bridge. The myosin head pivots to show the low energy state, releasing ADP and moving the thin filaments over the thick filaments, the power stroke. ATP binds to the myosin heads, causing the head to break free from the active site on the actin. The myosin head becomes energized as ATP is converted to ADP. One moment, please.
ADP.
And now we have the ADP. Calcium is pumped back into the... What is SO? And attached to the calcestrine, allowing tropomyosin to once again cover the active site, resulting in the relaxation of the muscle. How do I stop it? Okay. This is the sliding filament mechanism of the muscle contraction as it occurs in two adjacent sarcomeres. We have one they're adjacent to each other. We have the I bands here, the light colored, and the dark color are the A bands. And then you have the Z line, which represents the red line that you see, and the M line, which is right down the middle. And so here we have a whole fascicle, which is comprised of several different microfilaments, and inside are the individual fibrils. As you can see, these do happen to be edible. Moving forward. As you can see, there are actually pieces of fruit roll up in here. This darker orange color is going to represent the mitochondrion. And then we've attached a pipe cleaner here on the end just to pull out so that you can see that there are actual smaller microfilaments within the whole fascicle.